All right, we are going to get started. Thank you so much, all of you, for coming to, I'm going to make sure we're recording. Perfect. For coming to the Citizen Science Data and Earth Science session. This session is sponsored by the Data Stewardship Committee, and we'll have a range of speakers talking about citizen science in the earth science classroom, in earth science research, and earth science data management, sort of across the board. So it should be fruitful. It'll be set up with a speaker and then a quick time for questions afterwards. And we're going to start with the outgoing chair of the Data Stewardship Committee, Ruth.
So I think most of the people in the room know me. If not, raise your hand. Yeah, liar. Okay, so I'm Ruth Dewar. I've been in the field for I don't know how many decades, or maybe it's I don't want to know how many decades. But I'm currently with Ronan Institute for Independent Scholarship because I got dissatisfied with where academia was headed. And our motto is reinventing academia. Yeah, I think I better because I can't see my screen. There we go. And over my career, I've learned a lot of things about a lot of different things because I sort of do random things. So, you know, oh. Better. And so there's what I'm going to talk about today. First is terminology really does matter, even terms such as citizen science. And then there is this whole thing about fairing and caring for data. And then I'll talk a little bit about my experiences with data in the classroom and at data@risk.org. So citizen science terminology matters is the first half of the name of a paper that I wrote with a whole bunch of people who are on the citizen science email list. And it's about basically the factor that every single term that you can think of has issues. Citizen is an issue, science is an issue, depending on what community you're talking to. And this is just a little cartoon from the paper that just shows what some of those issues are. And there's actually the citation down there, and you can see there's, you know, lots of authors. And it, you know, it's, I don't know, can everybody read those? Can anybody read those? Everybody, even the people in the back can read those? Wow. Okay. In that case, I won't, but I think some of these are pretty funny. So, particularly things like academic scientists, sounds like we have no experience of the real world. So, you know. And so part of the paper has what, you know, various and sundry projects that might fall under the citizen science umbrella, you know, might call scientists. And in all the cases, there are some caveats with those and reasons why you might not want to use that one in certain circumstances. And like, I mean, I should note that this only goes up to the C's. They were alphabetical. And this is also alphabetical, and it didn't even get all the way through the C's. So citizen, science, these are contested terms. And so the findings and recommendations that really came out of that paper are that there isn't a single term that is appropriate for all the contexts. And in any given project that you think is a citizen science project, you need to choose those terms carefully and explain their usage if you want participation. And really, the participants need to be consulted on all of this. And given the opportunity to think about how that terminology affects them and what they would prefer to be called. For example, on a CIS paper that I was working with on Indigenous communities, our partners were not citizens, they were sea ice experts, because they know way more about sea ice on a daily basis than any scientist alive on Earth, because they have to use it. Actually, it's going away, and that's a real problem, but that's a different issue. Okay, so how many people know what FAIR is? Okay, I should have done that differently. How many people don't know what FAIR is? Oh, there are a few people. Wow. Okay, I might actually have to spell that one out. How many of you know what CARE stands for? Wow, I don't see any hands. Okay, cool. So FAIR stands for findable, accessible, interoperable, and reusable, and it is sort of the new buzz thing that, people want data to be. They want it to be fair. In other words, they want to be able to find it, they want to be able to get it, they want to be able to actually use it with other data, and they want to actually be able to reuse it for whatever purposes they need. So what is CARE? Now, this came out of the Indigenous Data Governance Community, GIDA, which is a global community of Indigenous communities. And they had to add that it doesn't just have to be fair, but it has to be fair and care. Because care means that, the data is being collected and used for collective benefit, which includes those communities from which it came, to improve governance and engagement, and to be equitable. They want to have the ability to control their data. In other words, that their rights and interests are recognized, and that the data both informs and is informed by governance. There is responsibility. In other words, that they want to have positive relationships with the people who are, doing the research or having the data or whatever, and that it needs to serve their purposes as well. In other words, it needs to be a win-win kind of situation where they don't just get taken from, but they're also, learning about and getting from, and it's a two-way street. And then ethics, for basically minimizing harm and maximizing benefits. In other words, justice for future use, etc. Now, this came out of the Indigenous communities, but speaking as a person who belongs to a community in Colorado, not Indigenous, I have to say that I agree with all of these myself, and I would like this, for myself and my data. For example, I just learned that our regional transportation district just bought all of the cell phone positional data for all the people in the Denver metro area for the purpose of planning bus routes and things like that, which sounds totally innocuous. But then I'm thinking about it. You mean somebody knows where I've been and it's my city government for every minute of the day? It's kind of like, do I really want them to have that? Do I really trust them to have that? So these are things I think people ought to think about. And so I guess the lessons I learned from all of this is that, the days when scientists could basically do whatever they wanted with whatever data they got from anybody, that needs to be over. If you're getting data about people, etc., those people better be involved, and they better know what really is happening with that data and should have some say and control over that. You really need to think about the reciprocity issue, and make sure that the situation is a win-win for everybody, and that ethics and relationship development are kind of a key component here. So that was step one. Step two, actually. Okay, so for some reason that I have yet to understand, I was invited to teach the fundamentals of data curation at the Graduate School for Library and Information Systems at the University of Illinois, Urbana-Champaign for several years, and I did that. It was a required course for their graduate students. And me being the person that I am, I insist that you don't know anything about data until you've dealt with some. So I actually required all of my students to actually do a data curation project of some sort. And what I did was I belonged to ESIP at the time. And so I would just, put a call out to the ESIP community. Anybody got any projects for students to work on? And typically I had way more projects than I had students. Some of those projects were very cool. And the process that I used was very much like a process that's being implemented currently by the At-Risk Data Commons, which is at dataatrisk.org. And that process is basically people come in with requests. The advocate, who was me for these classes, basically looks at them and goes, this one's not going to be possible. I'll take that off the list. And then I, sort them from these ones require these kind of skills, and this one might be easy, and that one might be a little harder. This one requires a programmer, those kinds of things. And then the students would select them based off their interests and skills, and then they would go try to do them. And obviously, that could be embarrassing if they immediately talked to the submitter and had no clue what they were talking about. So I always ensured that they had to go through me and couldn't talk to that person until they demonstrated to me that they actually understood what the issues really were. And at some point, the submitter would approve the results of that, and life was good. So this is actually a process that's currently being implemented. The first two steps have prototypes at this dataatrisk.org group, which is sort of crowdsourcing data rescue, I guess you could call it. Now, in the classroom, not all the projects were completed successfully, though in many cases, forward progress was made that was useful to the originator of the request. But some of the projects actually were completed and completed well enough that they went back into the originating repository, complete with the names of those students attached, as part of the provenance record of people who created this pre-ASCII, pre-EPSIDIC, pre-BCD data and translated it into CDF format, for example, and created SPACE metadata. And the repository was really grateful and credited them for all of that. For example, AGU posters and things like that appeared. So what were the lessons I learned from that? And that is basically that the skills needed for any kind of data project vary widely, and the odds of any one person having all of them kind of low. But that given adequate support, most people, irrespective of their background, can contribute in areas that match their skills. In other words, I wouldn't disparage the idea of citizens, people, whatever you want to call them, being able to actually help with your data issues, because in many cases they can, as long as it's managed well. And, you know, I have a lot more to say because I'm old, but I'm not going to because I've just done a lot of varied things over my career. And so that was pretty much it. And I'll take questions. No questions? Oh, Bob.
Seeing the CARE principles, and I've heard of them many times, but I never seem to remember what they stand for. I'm just wondering if you see any difference between the needs that the CARE principles have versus the needs that you would have if you were conducting human subjects research.
They're very similar. In fact, a lot of the CARE principles came out of human subjects research on indigenous communities, for example. And for that matter, I think they say they came out of the indigenous community, but, you know, I really do think that any community probably wants them themselves. Okay.
Next, we'll have a virtual talk from Lourdes Vera, who is a PhD candidate and 2019 Switzer Foundation Fellow and a member of the Social Science Environmental Health Research Institute in the Department of Sociology and Anthropology at Northeastern University.
Hey, can you see my screen?
Not yet. Let me give you back the presenter control.
Oh, okay. Oh, it's funny you mentioned the CARE principles. I just wrote a paper on genomics and the application of the CARE principles in genomic research. If anybody's interested in reading that, or I can send it your way.
If you send it to me, I'll send it out to all the registered attendees after the talk.
Oh, cool. Can you see it now?
Yep.
All right. So thanks for the introduction. I am talking about both my dissertation research and the Environmental Data and Governance Initiative, also called EDGI. So as part of my dissertation, I'm part of the Wiley Lab that includes Professor Sarah Wiley and then Goran Smalley-Vel, Sarah Lerman-Sinkoff, and Colleen Langley, who have helped put together these slides. And so I'm going to be talking about a theoretical framework we call environmental data justice, which includes citizen science, or we also call it civic science, since just like Ruth mentioned, there are many meanings to citizen, and especially when we work with immigrant communities, you know, we're working with people who are not technically national citizens. And also the civic sphere that idea really promotes transparent and open science. So I'm going to be talking about this at both national and local scales and presenting some questions about how to connect and bridge those two scales with civic science. So what is environmental data justice? Well, let's start with the problem that we face, and we were already talking about accessibility. And for example, I made this this chart, and I would love any feedback on it about data storage and accessibility and use from a perspective of the community. So we see a lot of strengths and weaknesses for different kinds of data. And so for example, open data, that is usually, you know, it's great because it's open, it's free to use and download, but it might be difficult to find, interpret, and requires restructuring, especially when it comes to TRI, or toxic release inventory data, where it's self-reported from industry, but still it's free to access. So we have those paradoxes, a lot of those. So with EDGI, we're beginning to assess data format and web interface of the EPA's Enforcement Compliance History Online, or ECHO database. And we're working on a project to look at it, from this lens of accessibility and where the data go. So, and another example is a lot of proprietary data that are vital to studying environmental health, and you see that in the private data box, like formulas for potentially toxic chemicals. They're not available to the public or to researchers. And so that leads me to our response. And our response is this framework of environmental data justice. And since academia, science, and the federal government are historically based on the construction of science in white, male, and capitalist spaces, the voices of marginalized communities and individuals are often ignored in environmental research. So environmental data justice, EDJ, therefore argues that the systems of oppression that are interlocking, like racism, heteropatriarchy, ableism, and classism, have constructed experiences of environmental injustice and the environmental data that we work with. And moreover, research on marginalized communities, most of us, I'm assuming, come from this perspective that an environmental justice data study, for example, using government data from afar, is different from an environmental study designed and led by a community. So this is why my presentation will explore questions about how we can envision large-scale and small-scale environmental data infrastructures that can be adapted by local communities and address their needs. And if you would like more of an explanation of this, I can address it in the Q&A. So at a national scale, this is where I talk about EDGI. And we have grown to over 160 members, with probably about 20 core members. And we begin with archiving. So the first year after Trump was elected, throughout the country, throughout the US and Canada, we collaborated with Data Refuge to host data rescue events. And this is where we archived politically vulnerable data and information, like studies on climate change and the climate change website that was taken down. It was a federal website through the EPA, and it was taken down. And then at these events, we also mapped out the different federal environmental websites that had data that were at risk. And then this is how we started web monitoring, by creating a web crawler that would go through and look at and check for changes in the federal environmental websites. So we found so many drops of the mention of climate change, and then some different tweaking of language, for example, taking away the connection between fracking and saltwater disposal wells, and therefore frack quakes. So we see a lot of changes that way. And then we also interview longtime federal employees that have been affected by the current administration turnover and the dismantling of the EPA. And then this also leads to environmental data justice, where we take a step back and reflect on our work. We have reading groups, and we support projects engaging EDJ-like citizen or civic science projects. And we actually had a webinar where the Desi Lomber Rodriguez from the U.S. Indigenous Data Sovereignty Network presented on Indigenous data, and they were a big part of creating these fair and care principles. And so since you can't see me, I'm in the picture on the top middle on the left. And there we are. I'm with Kelsey. We're at the Data for Black Lives conference. And this slide just shows our other activities, like the data rescue event in the top left corner. We meet all on Slack because members are just distributed throughout the area, throughout US, Canada. And then here's a picture on the top right with our beginning EDJ brainstorming event where we're trying to envision alternative practices for caring for data, creating data, and different, maybe alternative political infrastructures for more democratic data practices. And this leads us to our envisioning of EDGI's values, and then these are the starting point for EDJ. And I'd like to emphasize the points of intersectionality and us trying to create otherwises for data that are anti-oppressive and engaged participatory knowledge making. So like the data rescue events crowdsourced this archiving of data on a large scale, and then participatory knowledge making also includes citizen science on the ground. And this leads us to the local scale. I feel like I'm running low on time, so I'm going to go through these a little bit quickly. I've been working with the Wiley Lab on developing a do-it-yourself, low-cost hydrogen sulfide sensing tool. And this sounds a little bit complicated, but it's really just photo paper that has a layer of silver gelatin, and it reacts to sulfur in the air. And for those of us who are wondering what is hydrogen sulfide, we know it as that rotten egg smelling gas that can cause a lot of respiratory issues and is neurotoxic. A lot of the equipment measures it in short spurts, and there aren't a lot of tools that measure its chronic effects, like at low doses, like that people suffer from headaches, nausea, poor memory, and stress and depression. And so our photo paper seeks to capture these chronic amounts of hydrogen sulfide, and it's actually it's visual. We can visually map it, which makes it especially useful for residents to identify sources of H2S and to share it with others, because a photo paper on a map provides much more easy to interpret data than, let's say, a graph or a map with just numbers. So this is an example of Deaver, Wyoming, where on a ranch we found, used the photo paper to find hydrogen sulfide coming from a discharge canal. And this is how we make it. We put the photo paper into a canister and then fix it in the darkroom after we put it out and cap it after 7 days. And then we use an X-ray fluorescence machine to measure the sulfur and the silver on the photo paper. And we use this, there's a picture of someone putting out the photo canister in the field right in front of a pump jack. I'm going to skip through this because it shows the different, just the prices of the different tools, and that, the photo paper is really cheap, $100 per canister, while the Jerome is $15,000, which the Jerome Meter is an industry standard tool for measuring hydrogen sulfide. So we're developing this with communities and for communities. So we use, going back to the X-ray fluorescence, we can calculate or estimate based on the sulfur and the silver values how much hydrogen sulfide there was in the air. And we just published a paper on this in Atmospheric Environment, and Atmospheric Environment X, which is an open journal. And we found really, really good and strong correlations between how dark the paper is and the sulfur levels, and then the hydrogen sulfide in the air. So then we can use this to develop a colorimetric scale that communities can use. And this allows them to not have to, send their samples out to get tested by an X-ray fluorescence machine, and that's like $25 per sample. And instead they can line up, we can line up the photopaper strips to the scale and then estimate hydrogen sulfide concentration. And so working with the media, this is in Saskatchewan, Canada, this data, you can't really use it in a courtroom or with regulators or as a regulator, but it has really helped communities come together and build collective efficacy, and then publicize their issues that they face, and then open or get more resources for much more for regulatory attention and more air monitoring. So this is a report called The Price of Oil, and they reported on our photopaper project that we put out canisters in Saskatchewan, Canada. And then we had some results, or people's lives were directly impacted by this citizen science. So you see in the bottom, Mr. Gervais, he has a facility right on his property. And after this report came out, the company that owned the facility, they came out and they did technological improvements on his on the facility on his property, and they also I believe they added an air monitor. And while it's while the facility is still on his property and he still faces the everyday dangers of having it present, we saw some more response and oversight from the company. And so this leads me to question validation. We know scientific validation has been developed throughout many, since the scientific enlightenment, and entertains questions for scientists like accuracy and rigor. But then I wonder about civic validation. What would that look like? And how can data also address community needs and support community advocacy? So in conclusion, I'm ending with some questions for you all. So how do we connect the large-scale data that EDGI is archiving and critiquing to this flexible local scale data? Like, we use satellite maps in our work with communities, but that large-scale data isn't as good and accurate as, let's say, a camera. Sometimes Another project has been a camera attached to a balloon to get that communities let off into the air to get more accurate images of their land. And then how do we design tools, data analysis processes, and gather resources for community needs? And then what benefits are there to small scale community-based data that don't exist, like I said before, with data from satellite images or large scale studies? And that's it.
We have time for one question.
This is Michael Goodman. I've just got a question about the photo paper, and I'm trying to understand how often does that paper need to be changed? Because I imagine that the paper was exposed for 1 week versus a month, that the detection would be altered or would have a different recording based upon the long-term exposure.
Yeah. That is a great question. So usually we put it out for 7 days, and that depends on exactly how much hydrogen sulfide's in the atmosphere. If we have an area where there's a lot of it, then we'll probably only put it out for a week. If we have an area where there's little, then probably 3 weeks. But the colorimetric scale that we developed is based on 7 days, 1 week. So it does get darker over time. And by having that in 1 week, having that scale be just 1 week, we control for time. And right now, only community members that put it out for 1 week can use that scale. So we're in the process of developing a matrix that shows the different colors and then the estimates of hydrogen sulfide as time increases, and then as hydrogen sulfide concentrations increase.
Thank you so much. That was awesome. Next, we have Brian Hostetler to talk to us about what citizen science looks like for education.
More of a walker and talker. Thank you, everybody. Thank you. My name is Brian Hostetler. I am an educator, and I thought maybe Alexis mentioned this yesterday, that we might want an educator's perspective on not just the efficacy of citizen science, but also some good reasoning behind doing citizen science. And my perspective is always the education side. So looking at what the people gathering your data are actually getting out of those projects beyond good data, beyond the sheer volume of what you get. There are a lot of different advantages to the people that are collecting that information, and it's something to think about. So we're going to go over just a couple of those. Which one? I did. Oh, there we go. All right, so this is going to be a little more touchy-feely, I guess, than the other sessions were. But when you were a kid, what did you think a scientist looked like? And you can answer these ones. You can raise your hand. Rada's make it up. But yeah. You're not gonna like this answer. That's okay. I'm not going to like it. My dad. You think it looked like your dad? Okay. Okay. Does your dad look kind of like me? Beard? Okay. That works. Did he have hair? Because I used to. Okay. That's a little bit, that's eerie, yeah.
Lab coat.
A lab coat, yes, yes. Anyone else? When you were a kid, yep.
I always saw people splashing in the water collecting specimens, so they always had on boots, the high top boots and the goggles to put their face in.
So the high top boots and the goggles, that's more rare. That's a good perspective though. Yes, sir.
Movie star good looks.
Yes, obviously. Obviously. That one actually proved true the entire time, so that works out pretty well. I mean, that's what way it goes. Quick question before I move on. How many of you consider yourselves scientists? See, there we go. Terminology. Terminology is very important. So we are all scientists. And when I Googled, I literally put this together last night, but when I Googled, when I Googled scientist, I Googled the words grown-up science, and that's what I got. Does that look like anybody's dad, maybe? Looks a bit like me? Something disturbing about these images, these are all white people. These are all, and also, what science are they doing? Probably some kind of chemistry or yeah, maybe biology. For the scientific crowd, they're like, yes, I know what that is. It looks like a scientist who is doing that type of research. How is that right and how is that wrong? Like how a lot of us, I kind of look like those folks. Yes, sir.
So it tends to overemphasize lab work versus field work.
That's why that perspective of somebody collecting samples in the lab was a little more rare. It overemphasizes people that are sitting at a lab desk working on something, and that's not most of your worlds. It was never mine, and a lot of the scientists I've worked with, that was not theirs. So that's how it's right and how it's wrong. If you ask kids what they think about science, you might get some of these responses. If you go to a middle school or a high school classroom and say, tell me about science. What do you think about science? And they're like, it's boring. No, it's so much fun. They're like, it's too complicated. It's like, I don't understand. I just don't get it. That drives me crazy. I just don't get it. Or this one always gets me. Like, it's not for me. I'm bad at it. Have you ever said, this, and I'm not going to make you raise hands, but if you've ever said, I'm just not a math person. No, you can't say that. If you ever said, but probably not in this group, actually. Said, good point, sorry, got to work on this. If you say, I'm not a math person, I'm not a science person, I'm not a social studies person, but it's very rare, I know, it's very rare that somebody would say, I'm not a reading person. I don't read. I don't know how to read, and I'm not going to learn, because why do I need to know that? That would be a much rarer opinion. So, and I did another Google search and said, scientific data or sciences, and these are the top 25 reports, and I get that a lot of this does seem really complicated. Like, these are long words. These are not 5th grade words. These are not 6th grade words. A middle schooler is not going to necessarily be able to pull this up and understand what these words are, and so they think it's not for them. And so that's what we have that's really the opportunity with citizen scientists to start to address. Just for grins, I also looked at kids science to get images of kids science, and I like this more, right? This is an improvement because at least there are people of color, there are both genders not necessarily equally represented, or at least more than one gender is well represented. But again, except for this little rock star right here, everybody's in a lab and they're all wearing their lab coats, and that is a really popular conception of science. Whoops, I did that. It's still right and it's still wrong. Okay, but what we forget is that we have an opportunity to give kids the chance to do real science, and not real science, but real science, not in the air quotes, real science. There are some hurdles though that come up to that, and some of these are things that you're presented with every day. Like, you have to have training to do real science. You couldn't do what you do. You probably couldn't have done in middle school, or you probably couldn't have even done in high school. You had, college for that. So it takes a bit. Why do they care what I have to offer? Why does a kid care the things, or why do scientific researchers care what I, as a kid or as a member of the community, have to offer to science? Has anyone ever felt that way as an actual researcher, though? Maybe? Like, this is what I'm doing. Why does anybody care? How can a whole class participate? You can't do what you do in a vacuum. Almost nobody is doing research by themselves. And Ruth, your paper was a great example of that, where there were 40 authors on that study. How can a whole class participate? The same issue happens with citizen science with kids in the classroom. Do we have the ways to be published? Would you do what you do if you never got recognized, if you never got to share the things that you do with the rest of the world, with the rest of the scientific world or the world in general? And can we give kids and educational opportunities that same chance? And then, how do I see myself as a scientist? We all struggle with that, and kids struggle with that as well because a lot of them just don't, and we have to figure out a way to get there. All right. As you're thinking about studies, you have to remember, who are the teachers in the room? I know we've got a couple of teachers there. There we go. So the teachers might tell you, and they probably have more to add to this. I'm speaking for every teacher ever, which is a bad choice. Our time is our scarcest and most precious resource. We don't have a lot of it. We have 27 to 30 to 250, depending on the class, number of kids that we are helping. And with a lot of time devoted to a project, and if it's an optional project, we're more likely to not do it. We're more likely to say, that sounds like a really good idea, and I think it would really be fun and informative for the kids, but we have tests to get ready for. We have other things happening. So as you're coming up with a study, as you're starting to think about using students in your data, you need to be aware of that and then think about, how can I make this simple for the teachers? If there's a lot of meetings, it's going to be tough. Webinars work better, but still. Every classroom is different. The data you get from one classroom will not be the same as the data you get from another. Different teachers act differently with their kids. Different kids act differently with their teachers. The same kid will be super engaged in maybe the STEM class and completely checked out in language arts. So there's a lot of variability in every classroom, and so your sample sizes have to be big enough to accommodate that, depending on what type of work you're doing. Also, we're professionals too. We can help. Teachers they do a lot of work and a lot of training, and we know our kids better than anybody, sometimes better than the parents. We know what is going to work with our particular students, and a lot of times we'll know something that can generalize around the entire school, what types of things that you can do that are going to help you. So call on us if you are considering this. If you're wondering, what are people doing? I'm sure there's a lot more. I know there's a lot more than this out here. These are just a couple tiny examples of programs that are happening. I think it was last year that NASA funded 6 new citizen science programs covering everything from lake monitoring to snow monitoring to kelp density to all kinds of other great things. And they're being done through different scientific research institutions, schools, but also museums. So I'm going to get into that just a bit. That's my other thing I've done for 20 years is informal education institutions. There are the LA Natural History Museum is using iNaturalist. If you've heard of iNaturalist, show of hands. Yeah, good. Okay. If you're considering citizen science, and this was I'm setting you up. If you're considering citizen science, you should be looking at iNaturalist as a great example of a platform that works very well to collect large amounts of usable data. Let's see, that's what LA County is doing, and they're actually doing research on urban ecology. They're looking at what kind of wildlife is happening in an urban environment, and a lot of people don't expect that. So the other cool thing about that one is it's being done through family groups. It's not being done at a school level. It's being done through families, and so they're really encouraging families to come out and learn together and explore together. And this platform is a really good way for them to do that. So it's another way to go. DMNS, the Denver Museum of Nature and Science, worked there for about 10 years, and they're actually collecting DNA samples. It's the only museum that has an active DNA collection and DNA accession in their collections in the world. So it's a pretty cool, at least it was. It's a pretty cool institution. They're doing some good work there. They're looking specifically at taste and how your genome actually affects your taste receptors. They've been doing they did a bitter one, a sour one, a salty one, umami one. They've done a bunch. So that's a great one. And that's an IRB nightmare. If you've ever thought about collecting, speaking of care, and looking at trying to get permission and trying to get samples of that size, they had something like 6,000 samples in the last group, so it's pretty cool. And then USGS Earth Sci Week, just that website there. If you go to that website, you're going to see examples of about 10 or 15 different programs that are happening, probably by people in this room. So I will save my breath on that one. Informal science, again, don't forget, science museums and science centers, they're not the same thing. So I've gotten that wrong, and I'm in the field. So I called a science center a science museum, and my wife hasn't let me live it down yet. So science centers are people, a little bit more educational institutions. Science museums are educational, but also do a lot more research on the natural world versus science centers. Zoos, there's a number of different, think of applications for zoos to work with biodiversity. Art museums, this one has to do, there's a couple museums in Germany and other places that are doing preservation work, and they're using citizen scientists to help with that. There are history museums out there doing work, before and after school programs. This is a good one if you have kids that have spare time and they can do something to help monitor, whatever you're looking at. Before and after school programs are actually not a bad way to go because they have those kids for a little while. Just took some pictures. Okay, and that is all I have for now. I'll be available if anyone has any questions, or if you're thinking about doing your own study. The teachers in the room have good perspectives, so if you saw them earlier, ask them. And again, I've worked with many different cultural institutions beyond schools, so if you have any questions about that, let me know. Otherwise, I'll stop. Thank you. Oh, I think we have a question. We're doing questions now? Perfect.
Very interesting talk. So I assume you're a science teachers?
Yeah.
Okay. So do you usually encourage your students to become a scientist in the future?
So the question was, do you encourage your students to become scientists in the future? And honestly, it depends. I think I encourage the love of science, and I think most science teachers encourage the scientific principles of questioning and understanding, because that makes a good citizen regardless. But I think that I do encourage the use of science in that way as a way to get to the next step. Some kids are more apt to get into that. They're more interested in that natural world, and for those ones I would encourage. But all right, question in the back.
Yeah, so you had that slide in the beginning about people considering themselves scientists and not thinking they're scientist enough. And it reminds me of like, am I a real programmer yet type of questions. I'm curious if you, I guess, how you would help students understand that they're not just playing around in a sandbox, that it is in fact real science. Because I feel like that's the easy, like, you can click through your screen all day or take photos for iNaturalist all day, but you might not feel like you're really a scientist still.
And that's a really great point, because I forgot to mention that earlier. So the question was having to do with when you are showing the kids science projects like this, how do I relate to them that this is really real science, that this isn't something, because in a normal classroom, you will do a experiment to find the results that you're supposed to get, right? Just like all of you are constantly doing experiments to, no, you don't do that. No, you're finding new things. You're finding out new stuff using data. And I think that's one of the big misconceptions that people have with school and science, is that you're just trying to find the same answers to questions that have already happened. And so when you're doing a citizen science project, the responsibility is shared. Researchers have the responsibility to come back and say, this is how your data is going to be used, because we need to know that as educators, because I don't want to lie to my kids, right? I don't want to sit my kids down and say, what you're doing is going to help science, and then not explain, and then have it not happen, or have those results buried somewhere so deep that we never understand where they are. And so iNaturalist does a good job of explaining that this data is being added to a larger database, and then there's mapping, and you're going to talk about iNaturalist, so I won't steal your thunder, but there's iNaturalist has good resource towards that. The Denver Museum of Nature and Science project I was talking about, you see how your data fits into the larger picture, and then we have anonymized samples of things and show the numbers of people that have done this study as well, and how that fits into what things look like. The problem with that one is that you can't pull that data right away. But it's very important to make sure that teachers understand how this data will be used and where it's going to show up. Because you're right, otherwise it's playing in a sandbox instead of building a road. And you really want to be able to make some progress in science. And it's an amazing opportunity for students to get to do when it's real. So don't fake it. All right, thank you very much.
I'll mention that Brian was gracious enough to let me draft him at the very last minute after our education speaker was taken out by illness. So she wasn't able to make it today. So thank you again for being so willing to be on the fly. Next, we have Dr. Rebecca Forkner, who is an associate professor at George Mason University in the Department of Biology to talk a little bit about her research and how she uses citizen science.
All right, so I know how many of you are scientists in this room. I guess we're all scientists with movie star good looks. Raise your hand, though, if you would also consider yourself to be a field biologist. Anybody actually work outdoors still? All right, yes, there's at least one person in the room. Thank you. All right, so what I'm going to talk to you about today is my field, which is biodiversity science. So I consider myself to be an insect biodiversity expert. I'll explain that a little bit in a minute, what I mean by that. But the reason that I'm here to talk to you is that I guess from my perspective, climate science isn't necessarily the big thing that we worry about. What we're concerned about right now is the insect apocalypse. Has anyone heard that term or in the news? All right, so we're familiar with this idea then that we are facing an enormous amount of biodiversity loss. My question is how can citizen science help us? How can we sort of combine the information that we have about climate change with this information on biological diversity? What are our options? Now, click on it. There you go. Thanks. I love images like this. So how many of you would see something like this or produce something like this as a course of your daily activities? Okay. So to me, this information is just beautiful. As a professor who's been in a rapidly increasing department, I'm asked to teach a lot of things that are outside of my realm. I study insects, but I'm the only professor that they would come across in their undergraduate degree that would teach them about climate science. So I love that people make these things. When I look at this, what I see is what I would effectively refer to as global, high-resolution, structured. Maybe you would also call this coupled atmospheric, ocean, land, surface data. So these are terms that you are all familiar with. Now, I'm going to date myself here a little bit, and actually there's some individuals in the room who are at least my age, so maybe I don't generally start a talk by denigrating myself, but I have a point to make with this. There's an enormous number of terms on here, and I actually got these just by looking at the ESIP website and reading through what people were going to talk about today. All of these are terminologies that have been developed or acronyms that are in common use now that were developed since I finished my PhD. Now, this is an issue, it's a problem for a couple of reasons. One, I know that as an educator, particularly at the college level, that anytime we invent something new, whether it's bioinformatics, whether it's the microbiome study, what that means is that there's now a new class that my undergraduates are going to be required to take, and we're going to lose something else. So as all of this develops and becomes a necessary part of science education, what we are losing is those individuals that are in biodiversity science, those of us who go out in the field and find those organisms and tell you how they interact with nature. So these things are great and wonderful. They produce products that I can really use in the classroom, but they're problematic in that they are things that we now are required to teach students. Now, they're problematic for another reason as well. And that's largely because most of these things are involved, if they're producing climate science information, with these global high-resolution information. And if we talk about biological diversity, what I do when I go out in the field, and my background is actually in pharmacognosy, so what I do is go out in the field and find that one tiny ant that has that sesquiterpene lactone that might win me the Nobel Prize for finally finding another cure for malaria. But it is highly, highly specific, and it is on an enormously small scale. Now, if we believe E.O. Wilson that the little things run the world, these organisms are things that are critical to climate functioning. So if we're doing a biological diversity survey, we are typically operating minimally at a 1 meter scale. It would be sort of a normal scale that we would take this, maybe at 5 meters. And we might replicate that study over a scale that's less than 180 square kilometers. And those of you who are geographers or geologists in the room might recognize that as the 7.5 quadrat maps that USGS produces. Now, I will go ahead and say that, I consider myself to be a biodiversity expert because one of the things that I did for one of my postdocs was to manage one of the longest and largest biodiversity inventories. That particular study has been going on for about 30 years. It follows the diversity of over 400 species. And given the scale of that study and the enormity that it took to complete or to continue to monitor that information, I have to be embarrassed by the fact that that study really only covered 3 counties in size. So with an enormous cadre of students and individuals helping me with that study, we really couldn't cover the scale that we need to scale up to talking about how these organisms are going to respond to climate change. Now, the other problem that we're having, besides the fact that we're operating at these really small scales, is that the technologies that are available for those of us to look at biodiversity are really limited. Again, I'm going to age myself here. If you may recognize what we're looking at here on the screen, there are some people with a little bit more gray hair than me. So you're looking at one of the first handheld GPSs. So this is one of the first Garmin. So if your lab could have one of these and you could actually geolocate and have that data along with your biodiversity information, your lab was way ahead of everybody else's. Now, when I went to Mexico, this is, again, I'm going to date myself, 1991, having a handheld Scion, phenomenal. So you could actually digitally enter that information and maybe also put some geolocation data along with that biodiversity information. Now, what I will say about that is these technologies actually aren't useful in biodiversity science. I'll explain why momentarily, but I want to make a point that clearly we've come a really long way from this handheld device. My students will take their cell phones in the field with them now and enter data directly into an app with Excel. So we've clearly improved upon this. The Scion has gone the way of the dinosaur, and we can actually, or we have rather, increased the resolution of a lot of these technologies that inform us about climate changes. So a satellite now might give you some information, not on the scale of 20 feet, but on the scale of as small, I've read, though I don't know in reality, as much as 35 centimeters. And that's a huge improvement. But while we can increase things like LiDAR that can see through the canopy and maybe give us not that saturation that we get at 6 levels of leaf overlap, it might be able, for example, to reconstruct what something under the canopy, a building under the canopy looks like. It really hasn't gotten, the technology hasn't and maybe never will get to the point where it can track this particular harvester ant that I was talking about that has that sesquiterpene lactone and look at its population dynamics. We may never effectively get to that scale. Now, the reason that I brought up those particular types, that Scion, for example, is that when we take these in the field, in reality, it doesn't matter if you have an iPhone with an OtterBox, it isn't going to survive in the field. So if any of you are involved in developing some of that top technology, it doesn't matter if you go out. We took every different type of digital device with us to Ecuador on one of my caterpillar collecting trips, and in every instance, you're on a nice little trail, it starts to rain, and you're in the middle of a waterfall. Every single digital device that we took with us didn't come back functioning. All right, so ultimately, it broke down to the fact that we were exclusively using write in the rain paper and a pencil, and that was all we could use. So what this effectively means then for us is that while we have really improved since I've finished my PhD, all of this technology that's talking to us about climate change, and I sort of have a check mark there on that first arrow, we can get at this 35 centimeter resolution, we can tell you what that particular habitat is, maybe what organisms are present, what particular tree, for example, in this case, maybe a lupine flower if we're in Colorado, we've been talking about Denver, and we could tell you that in 30 years from now, that particular location is going to have snow. But if we wanted to tell you the biodiversity and how that might change, so is that particular amount of climate change at this global scale going to effectively turn into a different type of flower and more rain, or are we going to see this complete loss of biodiversity? We don't know. And we can't scale down or up with that biological information that we have. Now, I'm going to go ahead and explain why I think this is critical. One of the things that I do when I'm teaching my undergraduates in climate science is explain to them, it's a novel concept, not to you guys, but to them, that disturbance isn't random. So if I'm going to predict a hurricane frequency or a fire frequency, that information, particularly if we're talking about changes in the fire frequency in the United States, which is critically important, maybe for Australia as well today, that this particular map is not just the physical or edaphic information. It is a forest or a woodland type. So the biodiversity matters to the function. So it doesn't matter if this map is phenomenally accurate at predicting where planting zones will be for our agriculture in 30 years. If we don't know where the plant viruses are going to go, how the soil microbiome is going to change, we're not effectively going to be able to predict whether or not our ecosystems will support us. So there are rather data out there. It's what I refer to and what sort of we refer to in the biodiversity information as big unstructured data. Now, the reason that I refer to it as unstructured, that we call it as unstructured, it is citizen science. So I'm using this definition here of civic science is this contributory participation via data collection and processing. And you are as familiar with a lot of these different types of things. You may not necessarily know that Reddit, for example, has been used to look at opioid abuse and treatment in terms of some of the different types of things that Alexis is going to talk to you a lot about GBIF and iNaturalist and the things that they're doing. We refer to it as unstructured because it is exceedingly different than the types of information that we might use in this sort of global climate change science. If we're talking about remote sensing, that information is structured. It's structured in the fact that every particular satellite may have the same type of sensing information. So it's telling you, for example, very specific types of information, an amount of atmospheric gases. A lot of these things are concentrations or amounts. Biodiversity and the diversity of organisms and how communities change, even the data, how much and what type you collect or how you express it isn't standardized. There's a lot of debates in the field about whether you use a particular metric or not. So it isn't something that has a particular structure, even in terms of the data that you record. And when we are putting it into these contributory, these civic or citizen contributory depositories, it is unstructured in the sense that it lacks all of the information that we need to talk about diversity. And so this has led those of us who are working in bioinformatics and biodiversity studies to basically come to the conclusion that despite having this proliferation of enormous number of online types of data repositories, that it's really not going to address these species diversity declines, this big unstructured data. Now, some of that tendency, or some of the sort of explanation for why we think that unstructured data, the blame tends to be put on scientists. The argument is that if I can't, I'm not even going to ask you how many people know care and fair. It would take me forever. We might actually have to go back to basic biology to explain all of these terms, but this is the terminology that I use that is necessary for me to talk about biological diversity. And so some of the blame is often put on scientists in that we're not really good at transcribing what we do to citizens or civic individuals. But that's really not why we're having problems translating all of this big unstructured data to a predictive science of what's going to happen with biological diversity. So what I'd like to do is walk, and I'm going to go through this, I'm just stream through this here so Alexis has time to talk to you about some of these projects specifically in detail. So I'm going to list some of the problems with this data. And what it's going to basically boil down to is that those of us who are involved in building these platforms or contributing data to these platforms need to find ways to turn the unstructured data into structured data. So I'll start with what the problems are. I'm going to only list a few, and then I'll get to what some of the solutions are. All right, so the first thing is that there's a large spatial bias in the data that is contributed. A lot of people are nodding, you kind of understand this. I just took this random clip. I work on red maple phenology, how it's changing over time. And I took this particular map just from our local area and started to look at, sort of total number of records. If you look at where the majority of the contributory data are in this map, what we see is that contributions about biodiversity information, in this particular case, the location of a red maple, These contributions are highest in areas that are not normal areas. They're urban, high density. We may be talking about landscape species that are not native to the region. And so they're not displaying the normal biological behavior, the normal amount of ecology, and they don't reflect normal abundances. And if we are to look by comparison at the number of samples that are in a protected area, so this is the Shenandoah National Park where I do a lot of my research. The national park system is one of our more protected regions. Then what we see is there's considerably less amount of information. Now, in addition to lacking data on biodiversity in normal or undisturbed areas, our contributions really are biased to where people are. Okay, if you're going to take your dog for a walk, if you're in your backyard, if you're planting something in your garden, maybe you went with a group on a hike, and in reality, what we see then is that people are not accessing private property. Makes sense. Nobody sort of really wants you on there. But if we look at where a lot of organisms are, and I use the Florida panther as an example, it's very easy to radio telemetry. If you're trying to radio telemetry capybaras in Venezuela, it's not going to work. So there's a lot of organisms for which we don't have spatial data. In this particular case, what we've learned is that 31% of the range of this organism, and therefore understanding its population density and changes, is on privately owned property. Now, in this particular case, we can radio telemetry something. You really can't do that with the bumblebee, although I'm going to show some RFID chips on those later. So we're missing an enormous amount of private land, and we are not sampling in areas where organisms are going to display normal behavior. Now, in addition, even if we were able, say, for example, we drive down Skyline Drive, another one of my favorite places locally, most individuals, so citizens, not scientists, can't go off trail, and they usually won't either. They would go on a structured hike. And so again, they're not going to be in areas that are behaving normally. And in particular, they're going to be in areas that are experiencing edge and disturbance effects. And I took this one particular photo here because this particular red maple is one where when I'm out in the Shenandoahs in autumn doing my research, everybody stops to take a photo of it. This red maple, because it's always very beautiful and it's in an area where it's surrounded by yellow trees, so it's very striking. But this particular red maple is showing sunlight-induced edge effects, so its phenology, its sort of temporal change, may not be representative if we were to go off trail. All right, so our citizens are contributing data off trail and again in these inaccessible areas, and generally they're over sampling things that are synanthropic. So I take students on a research trip to Costa Rica every year, and they're always like, oh God, we don't understand. How is it that you can remember all these tropical plants? Well, the truth is I don't have to, because it's unlikely in a week-long trip with students that I'm going to go far outfield. And what I'm generally running into is the same 5 common abundant species, coatis that are going to walk across the trail. And I don't need to know the entire diversity of Costa Rica, for example, to explain this. So synanthropic species that like to be around humans, or common species, are really the things that most people are going to encounter. I can generally, if someone calls me up and says, there's a green bug in my bathtub, I can often guess what it's going to be. It's going to be something fairly common. Now, the other issue that we have with these is that what people are contributing often is a photograph. This particular study, this was published in 2015 by a colleague, is looking at what I refer to and what we refer to in biodiversity science as cryptic species. This is the idea that something can be different, biologically different in DNA and identical in appearance. And if we look at this, and this was a globalized Yule coalescent model. It's a technology here, a statistical technique for looking at DNA data. What she found basically was that even in our really well-studied species, and I use the papilionid butterflies as an example, what we see is that as much as 50% or 1/3 to 50% can be cryptic. So things that look identical and are multiple species aren't going to be picked up in these data sets. And then what that effectively means then is that laymen are very less capable here of taxonomic identification. And that's why I brought up in the beginning that we're losing these biodiversity scientists, the people who are on the ground deciding whether or not these are actually species. And so I'm going to make this statement here that everyone's going to hate me for, and that is that species can't be identified by photographs. And I'm someone who loves the bugguide.net, and I like that my students can go online and find these images and try to do the identifications by themselves. But a photograph isn't a DNA. And so what we see is then the contributions that are listing species, even if we have in some cases with, for example, I think it's GBIF, you have to have at least 2 specialists have to agree on that species classification, we may actually be obscuring the large amount of biodiversity that does exist. All right, so again, this is one that in this particular case looks very different, but is one identical species, the same species that shows some phenological changes with the season. Now, the next thing that we see is that there's clearly a bias in the things that people want to study or want to photograph. And I bring this up because I am talking about the function here. The species here that is critically important in predicting, for example, wildfire frequencies is this pine bark beetle here on the right, and species basically with a large impact on ecosystem function. In this case, the number of records in GBIF is up here, 75,000 for a monarch butterfly, which isn't representative of most butterflies in terms of behavior. All right, so the functional species, ones that are going to impact the functioning of ecosystems and the biodiversity of ecosystems aren't there. This is just looking, for example, at fungi. There's only one record of the most important fungi for nutrient cycling in trees that's going to determine whether we make a shift from an oak-dominated system to a maple system. All right, now, finally, what we also see is that photographs and contributions to these data sets are at most presence absence data. Something is here or it isn't. It's not time series. An occurrence isn't a 10-year time series that lets me talk about whether or not an insect is in decline. The only way that we do that is through mark recapture studies. There's that RFID chip on the bee. These are things that allow us to talk about whether or not we are or aren't actually experiencing a decline. Now, so the things that are missing from these large-scale database platforms are the spatial information, the temporal information, the actual identity of the organisms, and information about their function. What is it that these species contribute? And in particular, I bring up forest, fire, and trees because these are the things that are critical feedbacks here for this climate data. All right, so how are we fixing this? The first thing, of course, is that every researcher has suddenly jumped on the bandwagon of using a naturalist or GBIF to create structure and use this to make publications. So clearly, individual scientists are creating that structure. This was this hilariously long and involved process that these poor researchers had to go through to take all of the disparate information from all of the platforms on the European fungal database and turn it into something that is now usable. Now, the answer, of course, isn't for all of us to jump on the bandwagon and do these types of studies, but when we do, it does tend to have a kind of a snowball effect. So what this has led to, for example, now is a platform for submission of fungi data in North America. This is the North American Mycoflora Project. And what has happened is that rather than try to get all of these museums and all this disparate data together, they've set up a platform that has the structure built into it. So there's data on how to start your project. There's tissue submission protocols where you send something to an expert so you can find out what the DNA is. So we have that identification, and you are sort of providing that to your citizen or your civic individual prior to that. Now, what I will say is that if you have a taxonomic bias right now, my big wish is that we all make whatever our one organism is that we really love the next panda cam. I'm telling you, this particular insect is cuter than any baby, bow bow, whichever particular panda we're watching at the time. If you like it and you photograph it, please put it up, create a need, and others will follow. All right, so this was a one that was on Twitter and now has some own YouTube videos. This particular wasp actually controls the population density of spiders, which controls the population density of some of the praying mantids, which in turn has an impact on grassland structure diversity. All right, so individuals are creating this, but what we actually need then is for those of you who are involved in academic research, maybe you might be mentoring a PhD student, you might be involved in a project yourself, is to scale up. All right, so 3 counties in Missouri isn't enough. What I would like for those of you who are involved in biodiversity research to do is get citizens involved in a way that allows you to do regional level studies. So what we see, this is a bumblebee mega transect study. It solved a couple problems here. Rather than just sample near the individual's university, they got citizen scientists involved. They piggybacked them on the permit of the researcher. So the citizens were allowed then to access the Shenandoah National Forest and the entire Blue Ridge. They sent out sampling protocols and collections and gave people envelopes to mail things back. So they're scaling up. And what this has done, and there's a couple of things that are piggybacked on this, this is Kate LaCroix and Ariel Firebaugh and her crew from the University of Virginia. What they've done then is to scale up what would be a normal PhD study that would have been a very localized biodiversity study to something that is now minimally at the state or multi-state level. So we now have regional rather than local information on biodiversity. And so they got citizen scientists involved. And in the case of Kate, what she's also been able to uncover is not just trends in the decline of mason bee pollinators within Virginia and Tennessee, but also the introduction of non-native pathogens that are then killing off some of our native pollinators. So she's been able to track that. So the scaling up gives us more of this spatial and temporal coverage. So incorporating citizens into your research is important. Now, what we also need to look for is novel ways then. Not everyone's going to want to interact with an academic. What we might actually need is individuals that still have some ability to do sort of unstructured contributions. So I find that geocaching is a great way to solve that. You can place that particular cache, that thing that person is supposed to find at a particular location out there with the data sheet that they're supposed to record. They can take a photograph and submit it. And then you have the geolocation information with a time series. People will come back to the same location and take a photograph or tell you whether or not a particular bird or plant is there over time. In addition, that geocaching can be combined with the types of methods that Kate did in her PhD, and that is handing out these bee hotels that allow you then to collect and track this particular biodiversity changes. All right, now Brian has talked to us a little bit about classroom. I want to end effectively by saying that I think Ruth is right, that the days when scientists could do whatever that they wanted with their data can't just sit in a notebook in your lab. What I would encourage everyone to do is not just to use these platforms to create scientific activities for students, but to require data submission as a part of that. So Ruth, thank you for that. But what I would do is encourage you then to not just incorporate this into your classroom, but have students understand that the regular contribution of data to an open source platform should be a regular part of science. And that is really antithetical. It's like I'm pained just saying that, because for me, access to my information and having the rights to publish it first, really, like, your ideas and your data are your credibility in science. But it is something that we're all going to have to do. All right, I'll stop there, Alexis, and I'll take some questions after Alexis has a chance here to talk to you about some of these specific projects.
All right, so I'm going to skip through some of this pretty quickly since we are just about out of time. But I'm going to talk to you from a bit more of a data management, a little bit of the data quality and sort of considerations with what is actually in citizen science data. So I'm going to breeze through first bit about the hat I wear as a research associate and environmental data fellow with the Mohawk Preserve, and then talk to you a little bit about my ongoing research into occurrence records and image data for key species. So some of you might have heard me talk about Mohawk Preserve amphibian breeding data set at the summer meeting. In short, it's a data set that was paired together from a long-term legacy collector who, if you were in the last session, he was one of the original NOAA weather station observers who collected tons and tons and tons and tons and tons of different types of data. He primarily collected them, however, as note cards, which If you have worked with digitization, note cards written in cursive is probably the most difficult thing there is to work with. So the digitization process for these note cards was really time intensive. We had a group of retirees that came into the preserve pretty much daily to scan note cards. And then we started with just the relevant note cards, just ones that had salamanders or key species we were interested in, but quickly, quickly is relative, expanded to all of the note cards. It's a room probably larger than this one that's filled with just his documents and note cards and written results. And so we used not only the in-person digitization citizen scientists, but also Zooniverse Notes from Nature project. No, thank you. Is it there now? Perfect. So the Zooniverse Notes from Nature project allows us to have access to crowd transcription services for these projects. But it's also, oops, now I'm stuck. And so it's also an ongoing monitoring project at the preserve where we utilize classroom volunteers and preserve volunteers as well as paid staff to go out and monitor ongoing changes in water quality, weather, and amphibian breeding ecology. So one of the challenges with working with long-term citizen science data is that you have really different data formats and really different protocols through time, but it is still data that you want to have useful and in a similar system. So our harmonized data set had over 2,000 sampling events and over 150,000 individuals collected or analyzed over the course of up to present actually. So this is super valuable data, but making it usable and appropriately making this data credited to the collectors and the various people who have helped make it possible is really difficult. This data set is available, can't quite see it, on the EDI repository and has yet to be published on, so it's pretty new though, so I'm not too worried. One of the things we were able to discover about this data that's relevant to the community of users is that there have been really significant changes in the pH of vernal pools. If you're not familiar with the vernal pool, it's either runoff or rain-fed temporary pond, basically, that are really key in amphibian breeding ecology. So across the whole preserve, there was a significant trend in an increase in pH since the 1930s when the study started or when the data collection started. But some ponds, this is a single site, had a much more significant increase over that same time period, going from about 4 to 5.5 almost. So this is an example of data that is now being acted on. So members of the preserve and staff at the preserve are taking measures to continue actively monitoring these sites, monitoring rainwater pH, and considering management efforts to protect the species that breed in these vernal pools. So some of these challenges that we face as someone who's providing data and providing metadata is how do we appropriately attribute and credit the people who are creating these data sets. You know, as a scientist, if I'm collecting 150,000 individual records, I would expect to be credited in publication. However, in a lot of these citizen science data sets, it's typically the institutional aggregator that gets the credit instead of the individual collector. And doing that ethically, if you were to give credit to each of your individual contributors, can be really, really tough. You know, with a lot of citizen science, you're working with vulnerable classes. And if you're giving something like location data, what you might be revealing is, you know, the location of a 7-year-old every day for 6 months. And that's something that you need to be cognizant of. And additionally, working with some of these long-term study samples, you might not be able to get appropriate consent due to the collector being deceased. So you might not be able to get consent for attribution in a way you might hope when reusing this data. But these are really great opportunities to diversify science, as has already been talked about, in terms of abilities, age, background, scientific degree, community type. And you can provide great skill building opportunities across the data management lifecycle, as has already been touched on. You can also use these data products to help support a community in the questions they're interested in answering. iNaturalist has already been covered. I'll just share some other things that we've found in our research findings. This is one of my favorite findings, is that the educational level of an observer impacts what they find, but not necessarily how much they find in general. So looking at elementary, middle, high school, and college students collecting information on iNaturalist, sorry, my title's got a little messed up. The biodiversity of total collections is the same, but younger students are more likely to find things that don't move. So they're more likely to find plants, whereas the older students actually don't look for plants, and they look for things that are moving or things that are weird. They're more likely to find things like fungi, but that doesn't necessarily mean that one's better than the other. That ends up meaning that they're complementary data sets. So we're getting important and useful information from different types of users. On the instructor education value, but also as we were talking with data quality, how you train your observers can really impact the type of data quality that you get back. This was a finding from some research we did on students collecting phenology observations for Nature's Notebook. And as you might expect, the number of incorrect answers for what the phenology of a plant was decreases the more you train students. But it's very different based on the species they're observing. So this is just 2 species, but it has a really significant impact on this non-native elm, but not much impact at all on the common maple trees that students see a lot. So training impacts, but training is also modulated by maybe what they're looking at, what the protocols are. And it can be difficult to track through time how different users are trained or who trains them through their whole time as an observer. Some comparisons between GBIF, which are typically professional collectors, and iNaturalist, typically volunteers. This date right here, these days right here are Earth Week. So you can see some things that come out in the data set as on certain days of the year, certain times, you have different amounts of observations. This is one of my other favorites. If you're collecting during the week, it's probably your job. If you're collecting on the weekends, it's probably not. So there is some temporal differentiation between volunteer versus professional for data contributors. Spatial as well. So this is like kind of a population map, but then you also see it's also a tourism map. So these are red spaces are where GBIF is more likely to be an observation of a bee, where the blue ones are iNaturalist. And it does look like a population map, but it's a population map with a little bit extra. So combining data sources, citizen science, and professional observers are complementary. But understanding the different biases in your data set is key to not letting that carry through to your research output, or appropriately balancing your data set between types. There's also some taxonomic differences. So iNaturalist, you're gonna have lots and lots of bumblebees in the bee records, but GBIF is much more even. So professional collectors might be more likely to give you a true assessment or a closer to reality assessment of the biodiversity in a region, whereas iNaturalist might give you a better assessment of the abundance of species in a region or in a certain locality, which can be useful depending on the types of questions you're trying to answer. But if you're trying to answer a biodiversity question with iNaturalist, you might not get the right picture of reality. And alternatively, if you're trying to answer an abundance question with GBIF data, you might not quite get what you're looking for either, which is a problem since a lot of our declining species big papers from the last decade or so use professionally collected data, which doesn't always reflect abundance values. So this is just that again, a little differently. So the takeaways from this is that there are some challenges. Providing the relevant collector characteristics is tricky and could lead to unsafe situations. And quality metrics are tough too. So metadata for citizen science doesn't always include the training types that individuals go through or who taught them to observe, but it might need to in order for us to have a better sense of what the value of that data is. But as before, there are amazing opportunities for the use of citizen science data in research. using diverse data sets and diverse collectors gives you a diversity of perspectives that can be useful when you're trying to get a full picture of the trends that are happening across the earth, across biology, across biological sciences and ecological scientists. So there are differences in what is being collected based on who you are, in short. So that's about it for me. One of the things I'd like to leave you guys with is what are the roles that ESIP can fill? We have a community data cluster and the data stewardship committee, which are both, and actually the information quality cluster, which are all actively thinking about these questions and considering what does this mean, what does this look like? But there are places where I think ESIP is really well posed to step in to assist and support in development of things like best practices and standards. And had a lot of support from a lot of people, so I have a lot of people to thank. And thank you so much for coming to the session. You're welcome to stay. We're a little bit over time, so you're welcome to head out or you're welcome to come up to the front and to ask Rebecca or I any questions.